Now we'll talk you through the type of data that we have generated in the laboratory from your genes. Now you will find this kind of section in every one of the sensors, and it will essentially tell you what genes we have tested, what genetic variations we were looking at, what your genetic results were, and a quick overview if this is beneficial or negative for your health. Now, there is no need for you to understand all of the technological aspects of this. It is just meant for an external scientist or a doctor who knows about genetics to look at this data, and he can then understand what we have done, what we have found, and how we came to the conclusions in our interpretation. So, in the symbol section, you will essentially find a short name for the gene that we have analyzed. And these are usually quite complex names. For example, TCF7L2 is transcription factor 7 like 2. So there's no need to remember the name. It just shows you we have analyzed the different genes in the course of this analysis. Then the RS number is a kind of catalog number for the genetic variations that we looked at. You have to imagine it like this. If we, for example, look at the TCF7L2 gene, you can imagine it as a long string of genetic letters. And the whole genetic code is A, C, G, or T, and these four letters come up in different orders in this gene. Now, if you compare this genetic code from one person to another, you will find that it's almost identical. So all of the letters are identical. There are only sometimes small areas in this gene where there can be a different genetic letter. And these locations where some people have one genetic letter, other people have another, this is what we call a genetic variation. And a genetic variation has a kind of catalog number, which allows one scientist to talk to another scientist so that both of them know what kind of genetic variation they are talking about. And this catalog number is called an RS number. And you will find this here. So essentially this is a unique identifier for the genetic variation that we have looked at. If you look in the polymorphism section, this becomes a bit more technical, but usually what you should be interested in is this C to T conversion or A to G conversion. It essentially means if there is a C at this position in this gene, the gene works normally. This is what we call a wild type. It's the most common genetic letter in this position, and if there is a C, usually that's not a problem. The genetic variation can then, however, mean that there is a different letter at this position, and for this genetic variation, this is usually the letter T. So if you have a T, that means the gene is disrupted, and if you have a C, it means it works correctly. Now, for most genes in our genome, in our cells, we have two copies. One we have inherited from our father and one from our mother. This means from the TCF7L2 gene, we actually have two copies. And we can have one genetic letter on either of the two genetic variations. This is why here you see a C/C. This means the first gene has a C in the position and the second gene also has a C in this position. It could, however, also be a C and a T, or it could be a T and a T in case both genes are disrupted. So there's a lot of technical stuff here, but this is the raw data that the laboratory has generated, and from scientific studies we now know what C/C means for your health. In the column of OR, this is what we call the odds ratio, and this essentially means how much the risk of the disease is increased based on this genetic result. So as I've said before, C is the normal genetic type, so CC means not an increased risk because both genes work in the way they should, and this leads to an odds ratio of 1. You can also say it's an likelihood of developing the disease of 1. You're 1 times as likely to develop the disease, and 1 times as likely is just the normal risk. Now, if we skip ahead and go to the PPARG gene, in this case the person has CC. Here, in this genetic variation, this means it is an increased risk of disease. and this has an odds ratio of 1.38. This means this person with CC has a 1.38 times higher risk of developing the disease. So it's higher than 1, it's 1.38. So in other words, 38% higher risk of developing the disease. And if it causes a risk, you will also find the word risk in the risk column. Then it is also possible for genes to be protective. So you could have 1 or 2 copies of a protective gene. If we look at the interleukin 6 example, for example, this person has a G in 1 of the 2 copies of the gene and a C in the other of the 2 copies. And this is actually protective. So this person has a lower risk of developing the disease through this gene. So the odds ratio is a 0.91. So this person has a 0.91-fold risk of developing the disease. This means it's protective. This is why in the protective column you see the word protective. And responder means that this person, due to this genetic variation, responds to a certain thing in a specific way. It might be a better response to certain medication. It might be better response to certain preventive measures. And this kind of information you will find later on in the text. Now, since all of these genes influence our risk of developing diabetes, we will then in the end have to look at all of them together. So while one gene might not have an influence on your risk of diabetes, one might protect you from it, another one might increase your risk, and another one might increase your risk. Looking at all of them together will then in the end tell us your general risk. So you can clearly see you might even have a lower risk than the general population because many people have genetic variations like this, or you might have more risk variations than others and have a higher risk of diabetes. So this kind of table, as I said, you will find in all of the other sections. They might differ slightly, but this is the general idea of how to read this kind of table.